welcome to today's Development Presents webinar. Development Presents is a monthly webinar series which highlights early career researchers in the fields of developmental and stem cell biology. So today's webinar features two of Development's 2025 Pathway to Independence Fellows who work on gene regulation. Just as a way of introduction, my name is James Gahan. I'm an associate professor and group leader at the Centre for Chromosome Biology at the University of Galway in Ireland. And I was actually one of the members of the first cohort of Pathway to Independence Fellows. So it's a real privilege to be able to host a webinar with two of this year's cohort. So just before we get started, one technical thing. So if you want to ask questions during the webinar, please put your questions in the Q&A box. And then if we have time at the end, I will read out the questions. Okay, so today's first speaker is Anzy Miller from the University of Manchester. And Anzy will talk about how NGN3 oscillatory expression controls the timing of human pancreatic endocrine differentiation. So, yeah, we see your slides fine. So go ahead, Anzy.
Perfect. Thank you very much. Thank you very much for the introduction. So, yeah, my name is Anzy Miller. I'm currently in the Papalopoulou lab at the University of Manchester. And I'm talking to you today about neurogenin-3 oscillatory expression controlling the timing of human pancreatic endocrine differentiation. So the kind of big question that I'm trying to ask here is, how does a stem cell or a progenitor cell know when to differentiate? And I'm coming at this from a protein expression dynamic viewpoint because I think that this is crucial to understand cell fate decisions. So what do I mean by protein expression dynamics? So we know actually that protein expression can be highly dynamic. So instead of, let me just put on my laser pointer, instead of proteins being expressed either kind of on or off or on in a sustained manner, we know that a lot of proteins actually can be expressed in a much more dynamic expression with fluctuations and levels over time. For example, here, repeated peaks and troughs, aka oscillatory expression over time. And the question I'm trying to ask here is, can these oscillations in protein expression actually control the timing of differentiation? So the protein and the system that I'm interested in is neurogenin-3 in pancreas development. And if I take you through this schematic of the developing pancreas, so we have the pancreatic epithelium. We have some cells that turn on neurogenin-3, which are blue cells here, and they delaminate from the epithelium and differentiate into the endocrine cells that go on to form the islets of Langerhans. And neurogenin-3 is a transcription factor that is really important for this process. If you don't have neurogenin-3, you get no endocrine cells produced. And we know that neurogenin-3 as a transcription factor is really important to activate many of the genes required for this endocrine differentiation. So the question is, how does a neurogenin-3 positive cell know when to differentiate into the endocrine cells? And the system that I've been using is a human in vitro pancreatic endocrine differentiation protocol. So I start with iPS cells, differentiate through the different phases that resemble development until the endocrine progenitors that express neurogenin-3. This protocol, I can also go on to differentiate into the immature endocrine cells. So this protocol predominantly makes beta-like and pre-alpha cells, but I can get some of the other endocrine cells also from this protocol. So to be able to visualize and characterize neurogenin's expression dynamics, I need to be able to visualize these expression dynamics over time. And to do this, I created a knock-in fusion of the venous fluorophore into the neurogenin-3 loci using CRISPR-Cas9. So this is a homozygous knock-in creating a direct fusion, allowing me to visualize the venous fluorescence as a proxy for neurogenin protein levels over time. So I have these knock-in iPS lines, I differentiate to endocrine progenitors, and what I find is that neurogenin protein expression is highly dynamic. So if we watch this movie and this cell that I've circled, you can see that the expression level goes up, goes down, goes up, goes down over this time lapse. And you can see 6 example cells on the right here of different cells showing, again, very highly dynamic protein expression over time. And the interesting thing here is that this is not synchronous. If I replay the movie again, you can see that the other cells are fluctuating at different times. They're all, peaking and troughing all at different times. This is not a synchronous kind of expression dynamics. And we also know that this is not kind of a technical artifact. We have a nuclear marker, which is a constitutive scarlet expression. You can see that this is actually very stable in its expression. So we know these expression dynamics are real neurogenin 3 and not kind of a technical artifact. In the lab, we also have a statistical tool that assesses whether these peaks and troughs are actually occurring periodically and therefore can be classed as an oscillator. And we find that the majority, so on average, 81% of cells are passing this oscillator test. So the kind of conclusion here is that I found that neurogenin 3 protein oscillates in the majority of human endocrine progenitor cells, and the average period here is around 13 hours between peak to peak. So what is the function of this oscillation? So we know these oscillations exist, but what is the function? And to be able to ask this question, I wanted to be able to mess up with the oscillations. And we know from other work that protein instability is a prerequisite for oscillatory expression behavior. So I decided to try and mess around with these oscillations by creating a more stable version of neurogenin 3. And luckily, other people had shown that if you create a phosphomutant version of the protein, then you create a more stable version of the protein. So I did exactly this. So creating my knock-in homozygous phosphomutant version of neurogenin 3 protein. And indeed, we found that this protein was more stable and it did mess up with the oscillations. So instead of repeated peaks and troughs over time, we see kind of one pulse of expression that's taller, kind of broader compared to the wild type. And our oscillation test shows that fewer cells are passing as oscillators. So I've managed to impair the oscillations. Does this have an impact on the differentiation and on the cell fate of these cells? So what we find is that non-oscillatory neurogenin 3 affects the timing of differentiation. So at an early time point, we see increased differentiation to both main endocrine cells that we make in this protocol. And when we look at direct targets of neurogenin 3, and this is just, I've got 3 example direct targets in neurogenin 3 here, we're seeing increased activation at an early time point. So I show here, this is day 16 versus day 18. We have the wild type in the pink color, the phosphomutant in the blue. So the phosphomutant here is the non-oscillatory version of the protein. And we're seeing increased activation of these target genes at this early time point. Normally, it takes until day 18 for wild type to reach the same level. So we're seeing earlier activation of these direct targets, resulting in earlier differentiation to the immature endocrine cells. So if we have a look at the timing of differentiation, the wild type cells, the oscillatory neurogenin cells, we see the spread of differentiation over this window, over this time window, while the phosphomutant, the non-oscillatory, we're seeing earlier, more synchronous differentiation, which is kind of just illustrated down here that the non-oscillatory neurogenin 3, we're seeing synchronous earlier differentiation, while oscillatory neurogenin 3, we see more spread out differentiation over time. So we now know that neurogenin 3 oscillations are controlling the timing of differentiation, but how exactly does this work? And to kind of ask this question, we need to understand actually how the dynamics are decoded by its target genes. So we know that neurogenin 3 is a transcription factor, important for activating a lot of genes that are required for the differentiation process. So what part of the dynamics of neurogenin 3 allows for these target genes to come on for differentiation to happen? So if we imagine a oscillation over time of neurogenin 3, what part actually of this oscillation is important to activate downstream targets? For example, could this be the period of the signal? So this would be the time in which neurogenin 3 is present in the cell, being able to activate its target genes. Maybe instead the fold change is important, and this is our measure of amplitude. and we're measuring this by measuring the peak to trough fold change. So maybe the fold change is important, or maybe instead it's more of a levels-based decoding, where you need a certain amount of neurogenin available in the nucleus to bind targets, to activate targets, and only when there is enough neurogenin that in the nucleus to do these targets actually get activated. So to kind of cut a long story short, we found no evidence for levels-based decoding. So this is plotting the median level of neurogenin per cell and the time that the cell differentiates. So just kind of a schematic to show you here how this is what this is correlating. So the median of the oscillation trace over time with the time that the cell differentiates. And so you can see there's no correlation in the oscillatory or the non-oscillatory version of the protein. So it's not that the cells with the highest amount of protein actually are differentiating earlier. But interestingly, we did find a good correlation with the time of the maximum fold change and the time that the cell differentiates. So what this means is that shortly after a large peak, the cells shortly differentiate after that. So this is really interesting because this implies that the fold change can be read by the target genes to cause differentiation. But this doesn't seem very kind of intuitive, like how actually is the fold change read? And interestingly, there is actually a gene regulatory network that can detect and read fold change of the input. And this is the incoherent feed-forward type 1 motif. And so this motif has an input. So for us, this is neurogenin 3. It activates two targets, X and Y. And importantly, there is repression from the X targets onto the Y targets. And it's this combination, so this whole motif, that allows the Y targets to actually read the fold change of the input. And so this motif exists, but again, it's not very intuitive to understand how does this actually work. And this is where we turn to mathematical modeling to be able to kind of simulate these motifs to understand actually how this works. So I'm just going to take you through kind of two example setups. On the left-hand side, we have inputs with the same fold change but different levels, and then the right we have different fold change and the same levels. So I'll just start with the left-hand side. So we have neurogenin 3, which is our input, and we have two cells here. We have the red cell, which has a high mean level and a fold change of 2 times between peak and trough. And then our second input, our second cell, the black cell, has a lower mean level and the same fold change. And then in this model, we can have a look at how the X genes and the Y genes respond to these inputs. And this is the amount of X over time and the amount of Y over time. So as you can see, the X genes, they're accumulating over time, and they do read the difference in level between the two inputs. And this is because our X genes only has input from the neurogenin. So if there's more neurogenin, you will see more X genes. So the higher mean level cell, the red cell, causes more X to accumulate over time. Interestingly, though, the Y genes, they have exactly the same response, regardless of the change in level in the input. And this is because there is exactly the same fold change. And the way this works is that if there is more X produced, there is more repression onto Y. So the more input of the higher neurogenin 3 doesn't do anything to the response of Y because there's also the repression there on X. So this is showing how you can have different inputs with this, with different levels having the same response with Y, but how can we get different responses in Y? And this is by changing the fold change of the input. So if I take you through the right-hand side of the graph, we now have a black cell with a high fold change and the red cell with a low fold change, but they have exactly the same mean level. So because they have the same mean level, we now have a very similar response in the X genes, but now we start to see the difference in response in the Y genes. We have a much bigger response in the black simulation than in the red one. And this is because there is more time for the Y genes to respond to the large fold change in the input before it's repressed from X. So what we find from this model is that we have X genes that should respond to the level of neurogenin 3, and the Y genes that should be level independent, but actually be able to decode the fold change in the input of neurogenin 3. So this is great. We have a kind of model of how this works and how this motif works, but does this actually happen in our system? And interestingly, when we look at all target genes of neurogenin 3, they nicely cluster into two main expression profiles. And these are just some examples. So we have some genes that accumulate over time, which resemble our X-like targets that should respond to the level of neurogenin 3. And we have some targets that pulse in their expression, which is very similar to our simulation of the Y targets. And it's these target genes that should be level independent, but instead decode neurogenin 3 fold changes. And with some experimental validation, we found that our X-like targets did respond to levels, while our Y targets did not, which kind of our model predicted. And therefore, it's hypothesis that it's these Y targets that actually can control the timing of differentiation through reading the fold change of neurogenin's oscillations and could explain that spread of differentiation seen in wild-type cells. And so to kind of summarize what I've been going through, I have a human iPS-based system. I differentiate these into pancreatic endocrine progenitors, and I found that a transcription factor that's really important for endocrine differentiation has very dynamic expression. Neurogenin 3 oscillates in its expression in the majority of cells with an average period of 13 hours. I haven't gone into any detail, but we didn't find that the expression dynamics had a role in fate specification. But instead, we find that the oscillations were important for spreading out the differentiation by reading the fold change of neurogenin 3's oscillations. And this is capitalizing on the kind of variation of fold change over time in this expression window of neurogenin 3 to allow for cells to differentiate at different times, creating this spread over time of differentiation. And this is importantly decoded by a gene regulatory network, this fold change detective motif. And this work is recently out in Developmental Cell if you want to read more about it. So just for the last sort of minute or so, I wanted to go into some more detail about the work that I'm continuing on with. Because obviously this has kind of brought out a lot more questions to answer. And it's kind of along the ideas that we know that different parts we know that like the fold change, for example, for neurogenin 3 is really important for the timing of differentiation. But do any of the other parts of an oscillation, are they read by the target genes? So it does period have a role or level have a role, and how exactly do the target genes respond when you change these different parts of an oscillation? So I'm basically asking how do the different parts of an oscillation, how are the target genes reading these different parts of oscillations? And the way that I'm asking this is I need to be able to change these different parts of an oscillation independently. So I've been creating a tool that will allow me to do this. And this is based on a Degron microfluidic system. And this is based on the DTAG Degron system. So just to kind of go over this system, I have my neurogenin 3 venous protein, and I've also added this FKBPV epitope onto this protein. And what this means is that when the Degron drug is not there, this is expressed normally. But if I add the drug into the media, then this degrades the protein. And the kind of important part of this system is that this is completely reversible. So I can add and remove the drug at will to degrade and accumulate my protein at precise temporal, precisely. So I can create oscillations of different parameters to really assess what part of the oscillation is important for target genes. And so if I just show this movie of this in action, you see that we can decrease and increase and then decrease again and increase. So I can basically create oscillations completely from scratch and be able to add and remove the drug whenever I want at precise times to create different parts and different ways of an oscillation happening. So I have an example here of creating a short period, a long period, or sustained expression over time. And this will really allow me to unpick these different parts of an oscillation and really understand how and when the target genes respond to different parts of expression dynamics. And so with that, I'd just like to acknowledge everyone in the Papadopoulou lab, especially the people in bold that have helped out with the project that I've presented today, as well as collaborators, facilities, and funders as well. And thank you very much for listening, and happy to take any questions.
Thank you very much, Anzy. Very, very interesting. While we wait for people to type some questions in the Q&A, perhaps I can kind of start. So when you have these different X and Y targets, so presumably does each Y gene have a specific X that it's regulating? Basically, what I'm trying to understand is how complex is this gene regulator network downstream of NGN3 and
Yeah, really great question. Basically all the neurogenic target genes. So I've classed them as a target gene if they're bound by neurogenin 3 and they are activated when neurogenin 3 is present. So they have to be upregulated. And interestingly, this is around 600 genes, and they fall nicely into X and Y genes. You either get the accumulation like the X or you get a pulsing expression. But what it means is that I have like 300, well, I think it's 250 Ys and 450 Xs. So there's a lot of genes that are under the X and Y category. And I also don't know whether, so I know also looking at binding of the kind of, I know that there's a few X genes that I have ChIP-seq data from that I can see where they bind, and they bind a lot of the Y genes. So I know that maybe only a small number of X's are required to repress the Ys. But it could be, like you say, that different X's are repressing different Ys. Maybe one X is very good at repressing lots of them, but not all. I think this is, a very good question that is speculation right now that, unsure at quite how the gene regulatory network is going to, kind of work.
complex?
Complex.
Yeah, so we have some questions from attendees. So from an anonymous attendee, I'm not sure if it is different, but two oscillations with the same mean but different amplitude would have different accumulated signals above a given level. Could it be the accumulated signal over a period of time is what is being measured?
Yeah, so like kind of an area under the curve measurement where you're measuring the total amount of protein over time. So this also, we found no correlation between that measurement and the timing of differentiation. So we don't think that that also has a role, at least with timing of differentiation in this system. I think this is kind of part of my kind of future work, is to kind of understand how all of these different parameters actually can be read. And so that kind of measurement maybe still has a role, but at the moment we don't see an evidence for the timing of the endocrine differentiation for that.
So then Henry Bushnell asked, do you think the fold changes stochastic, or do they show any sort of pattern over time?
Yes, so we definitely see different fold changes over time. So there's definitely variation in fold change over time. Interestingly, we don't know how these different fold changes are generated, which is also a really excellent question to kind of work out how we're getting different fold changes over time. Is this just stochastic, or is there some regulation upstream? We don't know this yet. This is a great question.
Okay, and Naveen Ramakrishna asked, do you have any thoughts on the kinase phosphatases that influence the phosphorylation dynamics of NGN3?
Yes, so this is also a great question, because we know that the oscillations are at least in part generated through the protein instability. So we know that because the phosphomutant, we don't see oscillations. So the degradation machinery is basically creating the troughs every time to create those oscillations. And there's some great work from Anna Philpott that's been looking at how that happens. So the kind of the phosphatases that are essential for that. So there's some work that's looked into how that is regulated, and I think that's comes back to how are the oscillations generated is another great question, and I think that we know that the degradation machinery is definitely going to be involved in that.
Okay, and then we have one last question, which is it important in this system for cells to differentiate asynchronously using this mechanism?
Yeah, so this is also kind of an interesting kind of hypothetical question to think like, is it good for them to differentiate at different like spreading out that window of expression? Is that a good thing? And the way I kind of think about it is um, if you're creating these so that in the pancreas epithelium they migrate out of the epithelium and create these little islets of Langerhans, and maybe you need that asynchronous differentiation at different times to create lots of different smaller islets of Langerhans rather than kind of one giant one at the beginning, maybe. This is obviously, yeah, it needs to be, this is just me hand waving as an idea, but it needs to be tested. And a kind of another viewpoint is maybe you don't want cells all to differentiate all at once at the beginning, in case you need, um, does that mean that your progenitor pool is gone right at the beginning, and has no time there is no differentiation that happens at a later time point when it's needed. So it's kind of like, yeah, not depleting the pool too early and allowing differentiation to happen over a long window. We know from human development that endocrine differentiation or insulin producing cells differentiate over a whole 3 month period. So we know that this is quite a long period of time, which implies that maybe this is important. But yeah, lots of speculation there.
Okay. Oh, we have time for one more question. um, Zebrafish have a one central big islet, maybe worthwhile to check this cool hypothesis. So
Oh,
that's from
cool.
Willem Stals. So maybe
Yeah, thank you very
a
much.
comment rather than a question. Okay, I think we're basically out of time. So thank you again.